My name is Dakota Gutierrez, and today I'm going to be doing my informative speech. The title of my speech is The Past, Present, and Future of Cloning. I'm going to start my timer in 3, 2, 1. Claude Volhan said, "Cloning will enable mankind to reach eternal life," during a CNN interview after allegedly cloning the first human, a 7-pound baby girl named Eve. The purpose of my speech is to inform you about cloning in the past 3 decades and where it's headed in our future. This is important because technology has always been advancing. Cloning will impact each and every one of us. It might be that one factor that allows you to get an organ without a donor. This technology can help in the medical field, allowing humans to understand and prevent diseases. Cloning will be a part of our future, and every day scientists get closer to human cloning. I am credible on this topic because, just like many of you, science has always intrigued me. To prepare for this speech, I have done extensive research and I have spent countless hours sifting through academic and government articles. Chemistry and biology are some of my favorite subjects, and cloning is just something that has always interested me. Though I have never taken the nucleus out of an egg and then fused that egg with a cell using electricity and then placed that egg into a surrogate female host, I feel that I'm quite comfortable with the basic steps of cloning at least. I have split the history of cloning into 3 main points. Starting in 1996, I will talk about the first cloned mammal, Dolly the sheep. Secondly, I will talk about the 3 types of cloning and how they're used in this day and age. Finally, I will talk about the expectations for human cloning in our future. Named after Dolly Parton, Dolly the sheep, the first cloned mammal, was born July 5, 1996. After nearly 300 attempts of cloning a sheep egg, 1 attempt was successful, and thus, Dolly the sheep was born. Though it might be thought that Dolly's sole purpose was to be an exact copy of another sheep, history and philosophy of the life scientists stated the aim rather was to obtain genetically modified animals that would express in their milk proteins with therapeutic properties for humans. Like expectations for human clonings, scientists are trying to make an animal that is further evolved than their counterparts. But no matter how far scientists push the boundaries, Dolly died at the age of 6.5 in 2003 due to premature arthritis. What scientists had found from her death is clones may be like brand new organisms rather than copies of old ones. TheConversation.com stated this. Through reprogramming cells for cloning, scientists can neglect the aging process from the original host. Dolly's life was needed for advancements in cloning. Scientists were able to learn, adapt, and overcome problems they were faced in the early stages of cloning. Some of these advancements have led to the cloning of genes and entire animals. These advancements might also lead to the very first clonings of human organs. There are 3 types of cloning: gene, reproductive, and therapeutic. Each one coincides with one another while cloning an entire organism, but they each have their own individual tasks. Gene cloning is simply making copies of genes or DNA. But reproductive cloning is much like with Dolly, creating copies of an entire animal. And finally, the most important type of cloning in this day and age, therapeutic. It can create embryonic stem cells which can be used to clone organs. In the near future, organs will grow like plants because of the way scientists have used cloning, technology, and stem cells. For therapeutic cloning, stem cells are defined as cells with the capacity for self-renewal and development in a specialized cell that composes healthy tissue. DovePress.com stated this. The self-renewal property of stem cells is what truly allows cloning to work. Organs and other tissues can grow healthy tissues on themselves, allowing for cells to repair and restructure. Of course, cloning wouldn't be possible without all 3 types of cloning working in conjunction. Gene cloning gets the DNA ready to be cloned. Therapeutic cloning will transfer the DNA to the egg. And finally, reproductive cloning will determine whether or not the egg can flourish in the uterus and be born. Now knowing how all 3 types of cloning work together, we can look at a larger scale ideas for cloning and what is expected in the future, like human cloning. Weeks after Dolly the sheep was born, multiple governments banned human cloning in their countries. The latest update, as of 2008, stated that 70 countries had banned human cloning due to ethical, religious, and/or other reasons. The main difficulty with cloning primates and humans is spindle proteins. The National Genome Human Research Institute stated, spindle proteins are located very close to the chromosome in primate eggs. Consequently, removal of the egg's nucleus to make room for the donor nucleus also removes the spindle proteins, interfering with cell division. Other mammal spindle proteins are scattered around the cell, which makes having a nucleus donor easier. In 1999, the first primate, Tetra the monkey, was cloned, and scientists have been perfecting the delicate process of primate cloning ever since. With the advancements in cloning we have made, human cloning is technically possible, but it is a very delicate and expensive process. Nearing the end of our speech, I would like to review some of our main points and leave you with something to think about. We talked about the first cloned mammal, Dolly the sheep, and how her life impacted cloning forever, through new discoveries and genetic modification of a cloned organism. Secondly, we covered the 3 types of cloning and how they work to clone an organism, as well as technology that is developing to clone stem cells and human organs. Last and finally, we talked about cloning in our future and the possibility of human cloning no matter how delicate or extreme the process is. With new technologies like cloning, it is important to stay well informed. With new advancements every day and scientists' ambition for the use of this technology, cloning will be in our future. Cloning has been around for 3 decades and it's not going anywhere. Cloning is not what we make of it, but what it makes of us.